Welcome to Study Loop.
Hello students, in this question different bacteria were grown on different cultural media, and on the basis of that we are being asked certain questions. The first one here table shows the growth of these bacteria on MSA media, which is the mannitol salt agar, and based on the data the first question asked is which bacteria were able to ferment mannitol and which were able to survive in high salt concentration. As we know that the mannitol salt agar, it contains a high salt concentration and also a carbohydrate mannitol, and this media is both selective as well as differential, and this media selects gram-positive bacteria, certain species of gram-positive bacteria which tolerate the high salt concentration, while it discourages the growth of gram-negative bacteria because they cannot tolerate the high salt concentration. And among the selected gram-positive bacteria, it can differentiate between the mannitol fermenters versus mannitol non-fermenters. And in case of mannitol fermenters, we use a pH indicator, which is the phenol red, in which the color change occurs and the color produced is yellow if the gram-positive bacteria are mannitol fermenters. So if the growth occurs, that means the bacteria were able to survive in a high salt concentration. As we can see that the growth present here is only in S. epidermis and M. luteus, which means that both of them, S. epidermis and M. luteus, both have survived high salt concentration, while as we can see that the yellow color is produced only in case of M. luteus. That means the M. luteus ferments the mannitol. In second case, the bacteria were cultured using the MAC media, which is the McConkey agar, and this agar is also a selective and differential, and it selects and differentiates for gram-negative bacteria, while it discourages the growth of gram-positive bacteria based on high salt concentration. And in case of the gram-negative bacteria, it differentiates between fermenters and non-fermenters, as it contains a lactose. So lactose fermenters and lactose non-fermenters, and in case of fermenters, the color change here occurs from no color to red or pink on this agar, while in case of non-fermenters there is no color change. Now coming to the questions, the question asked is which bacteria were able to grow on MAC and which organisms were lactose fermenters. As we can see that all the three bacteria had shown growth. For the E. coli 48 hours, 72 hours and 96 hours, for all there is a growth. For S. epidermis and M. luteus, the growth occurs respectively for 72 hours and 96 for S. epidermis and only for 96 hours for the M. luteus. For all three show growth. While looking for the lactose fermenters, as we can see there occurs the red or pink color. As we can see in both these two cases, which is E. coli and S. epidermis, the growth occurs and there is a color change to pink here in both the cases and not in M. luteus. So lactose fermenters are E. coli and S. epidermis in this case. And in the last case, the bacteria were cultured using the EMB culture, which is the usine methylene blue. It is also a selective as well as differential media, and it is selective for gram-negative bacteria, and it contains a dye which is toxic to gram-positive bacteria. So they are being selected, and in case of the differentiation, it contains a lactose, and so we can differentiate between fermenters and non-fermenters among the gram-negative bacteria. So lactose fermenters, they form nucleated colonies with dark centers, while in case of non-fermenters there is no such thing. The first question asked is which bacteria were able to grow on EMB. The bacteria which were able to grow on EMB are E. coli and S. epidermis, as there occurs a very slight growth in case of M. luteus. Another question asked is what is the difference between. Another question asked is which organisms were lactose fermenters and how could you tell? So in case of lactose fermenters, we can see that E. coli, which has a growth is thicker and appears shiny and is dark pink, which means nucleated colonies with dark centers, as well as in S. epidermis, as we can see that there is a separate round areas that are raised and smooth with a purple tint within the clear streak. So these two are the lactose fermenters. I hope you found the video useful. Thank you.